Morning students, MSc Zoology Semester 4, Cell and Molecular Biology Specialization. Today we will study Arabinose Operon. So far we have seen lac operon and tryptophan operon. Both of the operon has been described in their structure and regulation both. This is the third operon. After this, the unit will be finished. Over. So basically today we will study Arabinose Operon. The figure which you are seeing is the structure of Arabinose Operon. Okay. Arabinose Operon. Arabinose क्या है? Carbohydrate है. More precisely, you should write Arabinose Operon in the levorotatory form. तो इसको लिखना चाहिए आपको L Arabinose Operon. Okay. So what is it? This operon, the L-arabinose operon, is also called the ARA. Italics में ARA operon or ARA small c and capital BAT, BAT. L-arabinose operon or ARA operon or ARA BAT operon, ये तीनों एक ही नाम है. It is the operon. It is an operon which is required for the breakdown of the 5-carbon sugar, L-arabinose. This operon is required for the breakdown of the 5-carbon sugar, L-arabinose, in Escherichia coli. ठीक. In this operon, there are 3 structural genes. You can see here, these are the structural genes, ARA-B, ARA-A, and ARA-T. That's why the name is here BAT, which is representing the structural genes. There are 3 structural genes. ठीक. These structural genes encode for 3 metabolic enzymes that are required for the metabolism of L-arabinose. These enzymes are ribokinase, isomerase, and epimerase. ARA-B gene product is ribokinase. ARA-A gene product is isomerase, a type of an isomerase. ARA-D an epimerase. ठीक है, नाम बहुत लंबा है इसका, तो शॉर्ट में epimerase. ठीक है, ARA-D gives rise to epimerase. तो मतलब जीन प्रोडक्ट कैसे हुआ, इसका ट्रांसक्रिप्शन हुआ तीनों जीन का, फिर ट्रांसक्रिप्शन के बाद जो मैसेंजर आरएनए बना, उस मैसेंजर आरएनए का फिर राइबोसोम के द्वारा ट्रांसलेशन हुआ. ट्रांसलेशन के बाद ये प्रोटीन प्रोडक्ट्स फॉर्म हुए. तो इन प्रोटीन प्रोडक्ट्स के फॉर्म होने के लिए क्या जरूरी है? ARA-BAT का एक्सप्रेशन. And the expression of ARA-BAT is tightly regulated by these control sites and regulatory genes. Okay. because when arabinose is available in the medium, that time only these genes will be transcribed. Otherwise, there is no requirement for the genes to be transcribed. Okay, suppose if there is high sugar in the medium, so sugar will be directly utilized for the energy production. So no arabinose uh, is required. No arabinose is required to give energy. No arabinose breakdown is required. Catabolism is required. So in the presence of high blood sugar, high sugar, also these genes are not transcribed. Okay, so that we will discuss later. First, you understand the structure of this operon. Okay. As a result of this expression of these gene products, ribulose isomerase, epimerase, conversion of L-arabinose to an intermediate of pentose phosphate pathway is generated. अभी आप जान लो ये तीनों जीन प्रोडक्ट L-arabinose को कैटलाइज कर दिया into a pentose phosphate pathway product, that is D-gylulose 5-phosphate. This is not a single step conversion. Action of these three enzymes result into this conversion that we will see. The structural genes of L-arabinose operon are transcribed from a common promoter into a single transcript. This is common promoter. सारे operon में आप देख रहे हैं structural genes का एक ही promoter है, common promoter. Here also you see common promoter. The promoter's name is parabad. P stands for promoter. Arabad is the operon's name. हो गया parabad. This is the operator for Arabad. Okay. And a single transcript messenger RNA is produced when it is transcribed. Structural genes are transcribed. When RNA polymerase sits over this promoter region. Okay. But the expression of L-arabinose operon is controlled by a single unit by the product of regulatory gene. The regulatory gene's name is RAC. कहने का मतलब हुआ इस अराबिनोज ओपरोन के एक्सप्रेशन को कंट्रोल करने में एक सिंगल जीन इन्वॉल्व है जिसका नाम है अरासी, एंड दिस इज रेगुलेटरी जीन. The gene product's name is RAC, which is a regulatory protein. Okay. So this RAC is regulating the expression of these structural genes. In addition to this, there is one more regulatory element which regulates negatively or positively this operon. The name is, you have already studied it, CAP cyclic AMP complex. In lac operon, you have seen CAP stands for catabolite activator protein. ठीक है, catabolite activator protein और cyclic AMP का complex, also this complex also regulates the expression of Arabide genes. Okay, अभी हम इसको डिस्कस करेंगे और डिटेल में जब रेगुलेशन पढ़ाएंगे इसका. जस्ट एन ओवरव्यू. अरासी जो होता है वो सेंसिटिव है फॉर द लेवल ऑफ एराबिनोज एंड इट प्लेज ए डुअल रोल. कैसे डुअल रोल किसमें है इसका? एज बोथ एन एक्टिवेटर in the presence of arabinose and a repressor in the absence of arabinose to regulate the expression of arabide. If arabinose is present in the medium, then this RAC will express these structural genes for the arabinose catabolism. If arabinose is missing in the medium, then this cyclic AMP cap complex, RAC is down regulating the expression of structural genes. So RAC ka role is important, very important in this operon. Okay, because it is playing a dual role as an activator in the presence of arabinose and a repressor in the absence of arabinose. Okay. साथ ही साथ RAC protein not only controls the expression of Arabide, but also auto-regulates its own expression. How? If there is high expression of RAC, if this regulatory protein is getting accumulated in the cytoplasm, then this high RAC level will switch off the RAC gene to stop further synthesis of messenger RNA and then translation of messenger RNA. तो मैसेंजर आरएनए ही रोक देगा ट्रांसक्रिप्शन, तो फिर ट्रांसलेशन किसका होगा? नहीं होगा. तो RAC खुद में ही सक्षम है अपने लेवल को रेगुलेट करने में, इसीलिए बोला गया इसको ऑटो रेगुलेशन. RAC protein controls the expression of Arabide as well as it auto-regulates its own expression at high RAC level. प्रोटीन का नाम में एक मिस्टेक हो गया, कैपिटल ए होगा, जीन का नाम में स्मॉल ए. ये प्रोटीन हुआ. Okay. Now understand it in more detail. You already know now that Arabinose Operon is composed of structural genes. This is the structural gene region and regulatory region. These are all regulatory regions. Control site भी regulatory region में ही है, regulatory region ही कहलाएगा. So this whole thing is यहाँ से तक इस रेगुलेटरी रीजन में दो ऑपरेटर रीजन है. कौन से? RAO1 and RAO2. ARA operator 2, ARA operator 1. ARA-O1 and ARA-O2. And there are two initiator regions, ARA-I1 and ARA-I2. सबका क्या फंक्शन है, क्या रोल है, वो हम अभी थोड़ी देर बाद देखेंगे इन द रेगुलेशन. ओके. In addition to these four sites, there is a cap binding site. Here cap cyclic AMP complex binds to this site. And there are two promoter regions, one promoter for Arabide genes, and this promoter para-C, and promoter for ARA-C is the promoter for ARA-C. You can observe here the direction of transcription is opposite. Here RNA polymerase will sit and transcribe from 5' to 3' direction. Here this as it looks like, but RNA polymerase can read the template from 3' to 5', but gives rise to messenger RNA in 5' to 3' direction. So here at this promoter, it will read the strand which is running in 3' to 5' direction. Here it will read the strand as a template at this promoter region. Here it will read 3' to 5'. So it will read a different strand. Here it will read a different strand, which is in 3' to 5' direction. Okay. यहाँ पर एक ही 5' to 3' लिखा हुआ है. सपोज इसको आप थोड़ा सा मॉडिफाई कर लें इस पिक्चर को. सपोज दिस इज अ डबल स्ट्रैंडेड DNA, देन प्राइम, एक स्ट्रैंड 5' to 3', दूसरा स्ट्रैंड एंटी पैरेलल है 3' to 5'. So this strand para-C, here RNA polymerase will read this strand which is running in this direction, and at para-bad it will read a different strand which is running in 3' to 5' direction. Okay, so here it will read because it will go in this direction. Okay, no, sorry. So this will go in this direction because it has to transcribe this regulatory gene. So RNA polymerase will go in this direction. So RNA polymerase will read this strand. Sorry, ये नीचे वाला नहीं, ऊपर वाला. थ्री प्राइम टू 5' को ये रीड करेगा, 3' से 5' डायरेक्शन में रीड करेगा, और RNA polymerase will give rise a strand of messenger RNA, give rise a messenger RNA 5' to 3' direction. Here it will read the second one, 3' इसका इधर है क्योंकि इसको इधर जाना है RNA polymerase को in this direction. So it will read the different strand as template 3' to 5'. Okay, now you have understood this concept. So these are the promoter regions. Cap binding site हो गया, and this is the promoter for ARA-C. This operator is for the ARA-C. O1 is the operator for Arabide, and these are the initiator regions, and this is the promoter region. तो आप यहाँ ऑब्जर्व करोगे for Arabide genes, operator is upstream to the promoter. Promoter यहाँ है प्रमोटर से डाउन स्ट्रीम में स्ट्रक्चरल जीन है, प्रमोटर से अप स्ट्रीम में रेगुलेटरी जीन्स है, रेगुलेटरी रीजन है, रेगुलेटरी साइट्स है. इनको जीन नहीं कह सकते क्योंकि इसका कोई जीन प्रोडक्ट नहीं है. But these sites are there. These sites are not transcribed. Okay. So, but here you can see पैरासी इधर गया होने, तो इसका ऑपरेटर जो है डाउन स्ट्रीम है टू द प्रोमोटर रीजन. Here the operator is upstream to the promoter. For RRC, operator is downstream to the promoter. Okay. There are three structural genes, ARA-B, ARA-A, and ARA-BAD. They encode the enzymes for L-arabinose catabolism. There is also a cap binding site where cap cyclic AMP complex binds to and facilitates catabolite repression. It represses the catabolite. Okay. It also results in positive regulation of ARA-BAD when the cells are starved of glucose. When glucose is absent, then it positively regulates ARA-BAD. So it is the role of the cap binding site. Because of this cap binding site, when glucose is absent, ARA-BAD expression will take place. ग्लूकोज का कंसेप्ट, अगर ग्लूकोज अवेलेबल है तो अराबिनोज को क्यों, क्योंकि ग्लूकोज इज इजिली अवेलेबल, instant source of the energy, because glucose directly go in the glycolysis और वहां से एनर्जी प्रोडक्शन होगा. लेकिन अराबिनोज को कैटाबोलाइज करने के लिए पहले ये 3 स्ट्रक्चरल जीन लगेंगे, उसके बाद जाइलुलोज 5 फॉस्फेट में कन्वर्ट होगा, ई फिर फेंटोज फॉस्फेट पाथवे के माध्यम से ग्लाइकोलाइसिस में एंटर करेगा. तो इसका पाथ लंबा हो गया. ये ग्लूकोज नहीं है तो मजबूरी में काम चलाने के लिए यह ओपेरॉन है, क्योंकि सेल मर तो नहीं जाएगा. ग्लूकोज नहीं है, अदर फॉर्म्स ऑफ compounds which can give energy that are available. So to utilize that, this is there. Okay. This regulatory gene RRC is located upstream of the L-arabinose operon. L-arabinose operon के upstream में है RRC. And it encodes the arabinose responsive regulatory protein RRC. नाम क्या है इसका? Arabinose responsive regulatory protein RRC. प्रोटीन का नाम कैपिटल A RAC. लेकिन जीन जो है ARAC, स्मॉल a RAC एंड ARA-BAD, इस जीन के प्रोमोटर्स अलग अलग हैं, डिस्क्रीट हैं. Where RNA polymerase binds and initiates transcription, the sites are separate. Here, RNA polymerase for ARAC, it sits on promoter for ARAC. And for ARA-BAD, it sits on promoter for ARA-BAD, that is para-BAD. ARA-BAD and ARAC are transcribed in opposite direction. Just I have told you. From ARA-BAD promoter, that is para-BAD, and ARA-C promoter, that is para-C, respectively. इन जीन प्रोडक्ट्स के क्या काम है? जीन प्रोडक्ट्स के काम है इस अराबिनोज का कैटाबोलिज्म को करना। कैसे करेगा? बाय द एक्शन ऑफ द एंजाइम्स. Enzymes are the gene products here. How they are performing? L-arabinose is the precursor which is available in the medium. Then Ara A converts it into L-ribulose. And L-ribulose is converted into L-ribulose 5-phosphate. Then L-ribulose 5-phosphate is converted into D-gallulose 5-phosphate. And in this conversion, catabolism, Ara C, these structural gene products are required. तो फर्स्ट कन्वर्जन एराबिनोज से एल राइबुलोज में कन्वर्ट होने के लिए एरा ए जीन का प्रोडक्ट which is isomerase, a type of isomerase is required. ठीक, इसका नाम है एल अराबिनोज. इस एंजाइम का नाम है एल अराबिनोज आइसोमेरेज और एरा ए. देखिए नाम याद रखना, एल अराबिनोज इसका प्रिकर्सर है, इसका आइसोमेराइजेशन किया तो एल अराबिनोज आइसोमेरेज, आइसोमेराइजेशन के बाद एल राइबुलोज जनरेट हुआ. So this is a type of isomerase, the first gene product which is required for the conversion of L-arabinose into D-gallulose 5-phosphate. So L-ribulose will get converted into L-ribulose 5-phosphate. Here you can see phosphorylation is happening at the cost of ATP. So kinase suffix will come. Enzyme's name is L-ribulokinase. Ribulokinase. This is Arabi. कैपिटल लिखो, एरा ए प्रोटीन है, एरा बी catalyzing the phosphorylation of L-ribulose to form L-ribulose 5-phosphate. The third gene product, which is an epimerase, RD, वो लगेगा यहाँ पर L-ribulose 5-phosphate को D-gallulose 5-phosphate में कन्वर्ट करने में. इसका नाम हो गया L-ribulose 5-phosphate 4-epimerase. L-ribulose आपका नाम है इसका राइबोलोज का आर छोटा से, एल राइबोलोज हाइफॉस्फेट। ये प्रिकर्सर हो गया, एल राइबोलोज 5 फॉस्फेट। और 4 ईपीमेरेज इसका नाम हो गया। इस पैरा डी। So this is the pathway where the gene products of L-Arabinose operon are required for the metabolism of arabinose sugar. So students, in the next class, we will see the regulation of this operon, and there we will discuss about these control sites and this regulatory gene RAC. How this whole regulatory region is involved in the regulation of these structural genes, in the regulation of the expression of these structural genes. Okay. Okay, students, in the next class, we will continue with the regulation. Till that, you digest it.